Okay, so welcome to the second session of the meeting, which has been beautifully set up by that presentation we just heard from Sarah and Aviv. And this discussion here is going to focus on a really important challenge moving forward, and that's querying the atlas. Because as we've just heard, clearly we're generating as a community a huge amount of data, with individual studies revealing the identity of novel cell types, how they relate to disease, illustrated very well by the COVID work. But as Aviv and Sarah both mentioned, we are now at this point of beginning to transition towards actually having an atlas. And we haven't maybe done that yet. We've constructed lots of individual maps of different systems, but do we really know yet how they link together? And I think we're really only beginning to learn how to query them. In other words, we're doing well at the data generation, but perhaps less well at constructing definitive atlases. And the aim of this discussion is to try and understand what queries would be of interest. And the motivation for focusing on the queries is that by understanding what people actually want, you can then construct the appropriate atlas. And so to begin with, I'm going to start by referring back to the Human Reference Genome Project, perhaps the greatest scientific effort across the wider community over the last 25 years or so. And that reference was generated from cell lines from a fairly small number of individuals. Other information, for example, population diversity, was then layered onto that reference. So projects such as HapMap, 1,000 genomes, etc. But the reference fundamentally, in particular in terms of the number of individuals that make up that reference, has remained relatively unaltered. And so when we think about something like the Human Cell Atlas, should a reference cell type atlas follow the same principle? Should we establish for each tissue a reference from one or a small number of individuals, and then we layer upon this reference information about differences due to ethnicity, age, and sex? Or do we actually need multiple different reference atlases that correspond to each of these variables? So do we need, pertaining to one of the questions that was asked previously, do we need an atlas that's different for elderly individuals as opposed to individuals who are maybe at an earlier stage of their lives? So we're going to start by thinking about this question, and I'd like to ask Fabian for his view on what sort of reference atlas we need.
Yeah, thanks, John, for the fantastic introduction. I think it's a bunch of fun questions to think about, both from application point of view, but I think also actually having quite a lot of implications from method point of view, how to actually do this type of integration and so on. I would like to start to briefly comment on this idea of one versus multiple references. And I guess it's a bit of a manner of definition what we actually mean by an atlas. If we now look on ongoing published paper, it seems to me that sometimes the notion of an atlas is rather wide, and your own particular data set potentially could be called an atlas. But I think in this discussion today, we would like to sort of have an atlas potentially be able to encompass a whole bunch of data sets that have to maybe taken in different labs. So for sure, an atlas should not contain any lab-specific effects, right? It should potentially be at least generalizable across a certain single individual. And the question, of course, now, as you rightfully said, it should be, if you have a small understanding of variation, should you be able to then just add others on top of it, or potentially just completely recalculate the atlas? And I think from a computational point of view, this really goes back to this question of in the end, essentially all these early processing steps are some type of question of machine learning, right? So how do we build this latent space that we then can query and ask questions on top of it? This latent space building essentially could be, we just sort of cluster each individuals, maybe we sort of batch correct them and then cluster them afterwards. But I think if we can make sure that these type of processes, as we in math say, commute, it doesn't matter which one you do first, if you end up always at the same thing, then maybe this question actually is not as complicated. I think in practice, we may not be able to make this fully independent, but I think having methods that sort of sequentially allow us to extend ongoing atlases, potentially not with completely recalculating everything, but just mapping existing annotations, clustering gradients, maybe existing queries on top of a larger data set without doing everything anew, either on just the user's side or maybe also sort of version Human Cell Atlas Lung 1.15 and sort of slowly keep growing that, but making sure not old ones do too strongly break, but sort of just steered into the right direction, maybe sort of defining a new subtype, maybe with a sort of hierarchical ontology and so on. Potentially we can talk about this later. I think will be the way to go. So I think you have to have flexible methods that sort of allow this transfer. And And later we'll hear from Rahul about one method we've been dabbling in a similar thing. So I think this will be around.
So Aviv?
I wanted to second what Fabian said, but also strongly from a user perspective. I think we all get excited as methodological people about the next great idea and method and trick and so on. And as scientists, by innovation. That's just why we all do what we do. But when you do research, there's something also to be said for a certain level of stability, meaning being able to peg your current findings to previous findings means that you kind of have a chain of understanding and discovery. And also you don't spend your time essentially in the end rediscovering the wheel or reinventing the wheel. And so that's one of the reasons that I find these reference-based approaches so compelling, as long as they give us the ability to find the new thing. And that was where I think the first generation of methods really focused on trying to peg the new to the old, or two things, and find their common denominator. Now we really appreciate the need to both find what's common and highlight the issues that are distinct. In the example that I gave on COVID-19, in one of the studies, what was done was that he took all of the existing annotations at a single cell level, not at a cluster level, and mapped cells to it, but it was also able to say, and these are the cells that don't fit. And in the cells that don't fit lie some of the most exciting things because they're unique to COVID-19. That is the balancing act that we should be after, and we should appreciate the importance of the invariant part because it also gives us a lot of confidence that these are real results.
Yeah, that's a really nice analogy. And I would like to turn to Emma now to think about how you think about this concept in the context of the Human Protein Atlas. Do you think that you need to have protein information from multiple individuals, or is one individual or enough to create a meaningful reference?
Yes. So first of all, I want to say that I totally agree with what Fabian and Aviv said. And I think that there is something to having this reference you can compare with and finding out of distribution, findings and other pin things to it. But when it comes to HPA, if you think of it, it's really not a great data set when it comes to comparing inter-individual or even comparing different proteins to each other. And still, it's a super valuable data set. So what I want to say is that no matter how we do it, it will be super valuable. And I think it's better to do something than to wait for the perfect. So in HPA, for every protein, we will have 3 different donors, normal tissue, 1 male, 1 female, if possible, minimum. That's it. And these donors, if we compare protein A with protein B, it can be completely different donors. Altogether, we have about 3 million images that come from over 7,000 different donors. But given this, we see very little inter-individual heterogeneity at the protein level compared to like diseased tissues where we see great heterogeneity. So it's a very small number of samples, and we cannot do a lot of intra-individual comparisons within the HPA data, and still it's taken us very far and given us a lot of knowledge about the human proteome. So I wouldn't be too afraid of going ahead without having samples matched from perfect individuals and separated over age and gender and all of that. I think we do need to capture some basics, gender and all of it for the reference atlas. But besides that, I would rather move ahead because there are so many things we will have to learn as we go on and so many challenges to solve. So if we're trying to design the perfect approach now, I'm pretty sure it's not going to be perfect.
That's a really nice point, Emma. Yeah, it's a really nice point that Emma just made. Yeah, and I agree totally. I also think while the analogy of the periodic table of cells has limits, categorizations can be really powerful in science, actually, in biology, and including in biology. I'm thinking about protein structures, and structural classifications of protein structures have had a huge impact on the community. And so again, I think for many reasons, with HCA's discovered so many nuances of cell types and so on already, even from the numbers of donors that we have, which is already not bad, in the hundreds. I showed for, and even getting towards thousands for some tissues and samples. Building these references in terms of whether you're talking about at the ontological level, whether you're talking about at the integration level, whether you're talking about at the machine learning modeling level, single-cell transcriptomes. They're incredibly useful for the community. And I agree that there'll be variation layered on top for sure, in terms of individuals and genetic diversity and so on, and aging. But yeah, nevertheless, I agree with Emma that having some sort of reference, at these different levels is empowering for the community.
So Shyam, then, following on from these suggestions, because I've got, for each organ, we have a male and a female reference, and that would be HCA version 1.0. Do you think that that's a suitable prospectus? As someone who would want to use the reference?
Asked to be provocative, so I'm going to be provocative. And also because I'm representing the Genetic Biodiversity Network, I'll say no, that's not satisfactory, right? And I'll tell you why. So let's take the human reference genome analogy that you so beautifully used to construct this all these questions. What has happened with the human reference genome? The original reference genome, as you mentioned, has been kind of a monopoly. Why is it a monopoly? Everyone in the human genetics field knows that if I have a Korean reference genome, that's much better to use for Korean genomics, for example, or a Singaporean reference genome is better to use for Singaporean genomics. Yet, everyone still uses the same old original reference genome. Why? It's not that people have not constructed a Korean reference genome. They have, a long time ago, high quality. It's because once you construct that, you're still stuck because that reference genome isn't annotated properly. So then you go back to going to the original kind of monopoly reference genome because that has beautiful annotations layered on top of it, right? So how can we address that challenge, not just of creating multiple HCA references, but also annotating them appropriately right from the start? And I'll just pose that as a challenge. I've taken up a lot of time.
Oh, it's fine. And I can see that Aviv and Fabian are competing both with physical and virtual hands to see who goes first. So I don't know who wins. Okay, Aviv, Fabian's conceded.
Okay. First of all, I want to strengthen what Shyam said. I think we cannot ignore the diversity of humans. I'll say the upside first of all, I will say that I think before things started to really get quick in HCA, to get, to become faster and faster and accelerate, there was a lot of concern that every patient's cells are going to look like a completely different thing. And we know now that that's not the case. In fact, as we've seen, for example, in the analysis in the lung, they kind of agree with each other quite well. And even in disease is not some complete craziness. It kind of repeats itself in predictable ways too. So it's not that it's impossible to put the diverse stuff together. At the same time, we know exactly for all the reasons that Shyam said, but in human genetics, and even more than that we have to accept diversity between humans as something that's going to have an impact on what cells look like. And it's not just because of genetic diversity, it's also because of experiential diversity. The same genome, meaning the same person with the same genome living in two different continents, is going to have possibly a different microbiome, is going to eat different food, is going to experience the environment differently. And as a result of that, their cells are actually possibly going to be somewhat different. So we really want to get the genetic and geographic diversity because geography really relates here to experience. And then finally, we learned from the last several years of advances in machine learning that if you train on non-diverse data, you end up with models that predict for the dominant population, no matter which population is the one that's powerful and reflected in your data, and you do not predict as well for others. In human genetics, that has been shown as well, most recently with the polygenic risk scores. We don't want to repeat this mistake. When people made this mistake in the 1980s and 90s, they could say that they didn't really know, but we know already, and we cannot repeat that mistake again. Fabian, your chance.
Yeah, I just want to sort of support that part. I think consistent currently we all get that, but I think Shyam has an important point, right? I mean, if we're not able to give same quality of annotation to whatever particular question you have, should that be in a particular geographic case, should that be in a particular disease situation, I think we kind of have failed, right? Because then, people come up with new names and sort of things get split up, it's going to be diverse. I mean, obviously, there's no way to sort of synchronize it and build this one unique ontology, but I think there's some ways. I mean, I think it has been mentioned before. I just want to highlight the cell annotation platform that Peter in particular, to myself, is most likely contributing to that, is developing. I think that's would be a fantastic way of sort of making sure that then the categories that Sarah was mentioning before will be a bit synced up, potentially in a hierarchical way, but being able to map across. Then I think the second part, and Aviv, you sort of brought up already, machine learning needs variation. We can very easily deal with that. We can also nowadays, we have those tools, which we didn't have, I guess, at this genome project, to know what can be mapped across, right? We can really transfer annotation, but then also transfer gradients and things like that. And we also know when it's wrong. So we have control of we have sort of uncertainty quantification methods now being generated. So I think from the method point of view, obviously there's still some challenges for that, but I think there's mathematical ways to define what's a consistent kernel up to a certain p-value or whatever, and the rest you can then also know what's individual specific, what's maybe geographic location, what's genetic specific. But then you can maybe also transfer additional things that maybe only particular labs can generate. Maybe there has been this super deeply characterized small subset where you have all kinds of omics levels. And maybe we realize that we don't need to do this full characterization for each particular geographic location, maybe a particular disease state, and then some of these things can be up to a certain level potentially transferred.
Yeah, so yeah, Emma?
I just wanted to say that thinking of it, one of the advantages of having a broader, diverse genetical background, for example, is that we can do targeted analysis only. For a lot of the methods that I work with, it's not genome wide. It's a lot easier to study 100 proteins than to study the entire proteome of cells. And if we know where the mutations are and what we think might be variable, which protein-protein interactions that might be changing, we know what to probe, and we can build much better targeted assays. So there might be a strategic kind of smart decision there, to a trade-off of doing a lot of work up front, but then allowing targeted assays instead of bottom-up assays further on.
Yeah, and I think for me, the thing that comes out from this discussion that's maybe unanswered is, are you still thinking about one reference? Or are you thinking for a tissue, are we thinking about multiple references? Because it could be one reference that which you layer on this other information and ensure that the annotation is good. And that reference could come from anywhere in the world somehow. And we just use that as the anchor. And then we kind of quantify the other cell types relative to that. Or are we thinking about multiple different reference sets as a function of age, diversity? Because I think this is really an important question to know. If you're clicking on the Human Cell Atlas in 20 years' time, are we going to be looking at one reference type per cell type, and then we layer on top of that? Or are we going to be looking at multiple different ones? And I'm still not quite clear there. So maybe Aviv first and then Shyam.
I will say that here, again, we allude to the analogy of the human genome, and I think it's very useful. When we conceived HCA, in order to not end up in with a problem that is entirely unbounded, as disease tends to be, we said that would be the healthy reference. There was also a question in the chat in the platform that Ellen will type an answer to in a second. Yes, occasionally we get disease samples in that context as well, including the fact that, for example, you might study immune cells which are healthy in the context of a tumor, which is a disease. But we did focus on the healthy reference. That does not mean that there wouldn't be disease atlases. And the analogy from the human genome is that you have a reference, and with all the caveats that Shyam highlighted, we should have had more than one reference, but that reference was of the genome just done in the context of the genome, not in the context of disease. And then you had many, many genotyping studies that focused on human diversity in the context of disease that led to GWAS. I think otherwise you would end up, you might use the same platforms and approaches and so on, but you would never actually be able to say, I have a thing. And so there will be multiple atlases. Some of them will be the atlases of disease, and some of them will be the atlases of the healthy body. Also remember, the disease might not be the whole organ. It might not be just one organ. It's hard to slice and dice. And as a result, I think we would have to leave it its own unique places, and the ability to compare and relate between them.
I mean, I'd be curious how you think something like HapMap, for example, would fit within the context of what you've just described.
Yeah. I mean, can I just add a point? So I was going to say exactly that. So Aviv made the point about the healthy versus the disease, and definitely we need to make that distinction for sure. But in terms of a reference, I think the comparison with the Human Genome Project, it really only goes so far, because nowadays, you know, thanks to lovely people like John and Fabian here, and you know, others, we've got fantastic machine learning methods that can actually describe a reference that encompasses multiple data from many individuals. And so I think a little bit we need to get away from this like one reference kind of mindset. We can calculate references that are already drawing on data from small populations of humans, you know, basically.
Yeah. So you can have a reference.
Others think about that, like
you have a reference of, I don't know, healthy adult liver, which is already a distribution across individuals with the ages and the different backgrounds and the different geographies and the genders, and everything is encompassed in it. And you might have another reference, which is hepatocellular carcinoma, a cancer of the liver. And by having the two and having the right approaches to compare things, not just at discrete levels, but also at continuous levels, you really understand how one relates to the other one. But having this more modular approach, I think, is both more realistic and pragmatic, but also fits very well with the differences between health and disease.
Shyam, sorry, I know you had a point.
Yeah, I wanted to thank Aviv for supporting the idea of multiple references. I'm a big believer in that. And I think to motivate this discussion, obviously having multiple references is more effort. It's harder for us and all kinds of technical challenges and so on, which we as computational people will jump at, but still it's a harder problem, right? And so then if we want to make that effort, we have to convince ourselves that it's worth it. And actually what's interesting is that even for healthy, okay, we know disease is different from healthy, but even for healthy transcriptomes, I think we're still in the very early stages of understanding what are all the axes of variation and how strong are they, right? How much does age influence our transcriptome? How much does gender influence or sex influence our transcriptome? How much does geography and ancestry influence our transcriptome, and so on? And the stronger that influence is, I would say the greater the effort we need to put into having multiple references, right? But at least right now, from what we can say, okay, let's say looking at ancestry alone, which we've done some very preliminary analysis on, we were really surprised to see how much ancestry influences the transcriptome. Even when you take out lab-specific effects, okay, you can analyze different ancestries in the same lab, we were hugely surprised to see how much variability we saw that's systematic across the ancestries. So that gives us some personal motivation that, you know, it's really important from the start to define multiple references and build our data structures and our methods accordingly. Once we go down that path of having only one reference, we lock it in because the whole ecosystem adapts to that single reference, and then it's hard to come out of that. You know, you're essentially already down one path in the Waddington landscape, right? So I think we have to take our first steps very judiciously. Yeah.
Okay, so maybe one point from Aviv, and then I would like to go into the next question, if that's okay. Oh, sorry, Fabian, do you want to go first quickly? Your turn.
Sure, it's okay. I just wanted to really briefly comment on that. I think there's really 2 points. I think from a purely pragmatical point of view, we see for pretty much every organ, we have already so many individuals around. So we won't be even able to agree on a single reference person. There's going to be many anyway, right? So there's going to be this variation. We just need to sort of really take care to make sure that we somewhere note down what the axes of variation are and potentially do some decomposition or whatever. So I think there will never be a single potential sort of just single reference. But of course we can do means, right? So I think from a pure user's perspective, it would be very attractive to say, hey, what's my mean expression, potentially variation, sort of width of that ball or whatever in expression space in that particular organ, this particular cell type. Because then I maybe want to use it to deconvolve my bulk or something like that, or do whatever type of downstream analysis. So I think just building in that we make sure we keep track where things come from and sort of communicate this, but then also have a projected simpler version of the overall, then obviously not just variants in cells, but also in population variants, is something I think users will definitely ask for. And I think that's kind of what's happening in some of these sort of small integration steps as well.
Aviv, and then we'll move on.
Yeah, so I think we're getting fooled a little bit by the fact that in the human genome, the reference was actually this invariant sequence. Yes, there was a little bit here, there was a SNP, there was a SNP, but generally it was just one long sequence. In our case, a reference is a distribution. There's even in one sample, you have a distribution of cells, but as you go across multiple individuals for one reference, you have this much more elaborate entity. And we're still learning how to think in those terms. But that should be our long-term goal. And I think that would directly address Shyam's point. That instead of this, here's one static entity and another static entity, and you can only perfect so many static entities, you have a distribution. Distributions require different computational tools and different approaches. This also addressed a question, I think, from Gamal Akabani, who is in the chat and asked, but should the reference be asexual? And then there would be, you know, here's the female, here's the male, here's this group, and here's that group. And I would say, no, what we're learning now with more advanced machine learning that we didn't have in the 1990s is that we can have that whole distribution and its many factors and how they relate to each other, both in, you know, stripping them each apart as well as their non-linear interactions. Fabian actually has beautiful work along those lines, and that's what it would be. And we would have computational tools and ways of reasoning within this kind of framework. That would be our contribution as a field that actually came with a new kind of data that had its own richness and properties, and it's more appropriate for describing entities like cells that are dynamic and change all the time. They don't actually stay fixed like our genome does, you know, up to somatic mutations. They change, and they change rapidly and slowly and multiple timescales, and we need to be willing to embrace them in this way.
Yep. So then moving on, so once we've constructed this set of references, what's going to be the equivalent of searching for genes regulatory elements? I appreciate that this is kind of going back to the human genome analogy, but a lot of work was done by finding new genes, on finding regulatory elements, a lot of computational methods developed to mine that reference, and a lot of biology has followed from those initial mining efforts. So what's going to be the equivalent in the context once we have different cell atlases? And does this vary if you're looking from a gene or a cell-specific perspective? So maybe Emma, if you'd like to start this, what would you think we're going to be searching for in the reference sets here?
Oh, I think right now, to start with, I think, you know, 99% of the Protein Atlas users are still looking for their favorite genes. So I think we will have to really build models and tools to help people to explore the data in different ways and see it through different lenses and really explore this multidimensional data that we have to kind of get, not only have the power users doing more advanced queries. So to the boring answer is that today I think we'll see a lot of, you know, how is my gene expressed across these different cell types, or what is this cell type, other cell type similar to, or cell states that are similar to each other, disease trajectories. So I think it will start out with quite simple queries like that. But of course, I would love, coming from an imaging perspective, I would love to be able to start to query the data from a visually similar, being able to find cells that are similar, to have a similar appearance in a microscope image to this cell, for example, to be able to really look at the data in different ways. But I think that we really need to build this framework for how to interact with the data, being even agnostically or with a certain disease narrative in mind, and allowing people with different backgrounds and expertise to collaborate and co-discover and co-explore in this space, because that's not easy. And I don't think we have the tools to do that yet.
Excellent. That's a really good answer. So maybe I'll turn to Aviv. Actually, you're unmuted, so by default, I'm going to turn to you. So what do you think the equivalent is going to be of searching for genes, regulatory elements?
So I think in the Atlas, we are used to searching for cells because we got very enamored with our cells, but it's just as important to look for the programs, so which don't necessarily fall into the neat categories that sometimes cell types fall into. And when you talk to a biologist, they usually have two immediate questions: where's my cell, where's my gene? And I would generalize from where is my gene to where are my genes. We know that that's a more robust question, a query. And we also know that usually the answer is more informative than just for an individual gene. So I would imagine these would be the first two queries that we have to contend with. And the second one on the genes is one that we have to pay more attention to, maybe because we got a little used to thinking about the cells. They'll be going backwards in time when genes were reigning supreme. And if we gave answers like those two, I think we will have done a lot for the community in terms of enabling every biologist at asking the right questions for the problems that they have.
Shyam, what do you think?
I think the question is about what kinds of queries we'll make it. Is that correct, John? I was just furiously typing into the chat to answer one of the questions.
Sorry to be a distraction in the chat, Shyam. Yeah, I think I can just chime in while Shyam is tapping, and then he can reply. Is that okay, John?
Sure.
Yeah. So I think the cells and the genes will be important, and we should also cater to as a community, to queries on programs like which cells are proliferating, which cells are undergoing oxidative stress. So these kinds of questions about biological processes, expression programs. Which cells are responding with an innate immune activation program, and so on in this sample. And then the other thing that will come, I think, is structures in tissue sections, like where are vessels, where are glomeruli, where are these kinds of sort of microanatomical structures. Is something we should start thinking about for HCA version 2.0, let's say, querying the atlas. And I'll stop there, and maybe Shyam wants to chime in now, or someone else.
Yeah, I think again, regulatory elements, genes, they're discrete units relatively, and more easy to count and so on. What are the units of a human cell atlas, right? Often, as Fabian started out, it's a continuum very often, but still maybe we can define some centroids in that continuum. So then, you can say, what are the cell types? What are some landmarks within each cell type that define some subtypes? Because ultimately, when you do functional assays, you don't want a distribution. You want, this marker is at this level sometimes, right? Make your experimental design easier. So can we define some landmarks in those distributions? And maybe those landmarks will be some units of inquiry, or some things you can download from that data set. And then if you take those landmarks, those are positions in functional space, positions in gene expression space. What anatomical locations do they correspond to? What functions do they correspond to? What other annotations do they correspond to? For example, if you look at a UCSC genome browser, you can take one gene and then click on it, and then you can get just a very long list of annotations for that gene, right? So ideally, we will be able to click on one place in gene expression space and get all kinds of associations and annotations for that, right? So let's say that's the cell type query. And then gene query would be, okay, what are the markers? If I have my gene, which cell types or which cell states have that gene as a marker? What is the distribution of expression of that gene across a tissue, across cells? Which genes are correlated with my gene? So that'll tell me what are the functional relationships and so on. I think these are some of the common questions people might ask. But of course, as we build it, I'm sure we'll be surprised.
Absolutely. Fabian?
We just discussed, like, this is sort of a matrix, right? Cell by gene, and we ask from this one direction, we ask from this other direction. But I think from the previous discussion, we also saw that there's sort of additional axes of variation. Essentially, we're not talking about a matrix, but about some type of hypercube, right? Which would be sort of layered also by people, would be layered by temporal, like, let's say, development, would be layered by spatial localization, things like that, right? So, I mean, you kind of hinted at that, but I think curing for these things and in combination, that could be, I think, what people will start asking. But then, and I think this is sort of turning it around, I would be personally very interested then to see what's actually shared. I mean, cross tissue, you were mentioning this briefly before, but even just, you know, these categories that we have now manually annotated and we pick up some gene ontology or so, right? Hopefully, maybe we can now come up with a data-driven version of that. Hopefully, we can come up with a some shared structure across maybe spatial temporal level that maybe is also shared across omics levels, right? And that could be maybe then called some regulatory element for this or that. And I think these data-driven ontologies, I think, would be a very exciting way to then sort of build additional things that people would then sort of try to look up in a particular data set as well.
Excellent.
The metadata is clearly very important for that, because there was a question about metadata before. And I mean, what Fabian says is totally right. And that's why having clean and as comprehensive as possible metadata on each sample when the data is deposited is so important.
Yeah, absolutely. Emma?
Yeah, so I agree with that. The spatial connecting gene expression and spatial information would probably be quite an early query that we'll see a lot of as well, trying to move between different assays. But then I was thinking of the human cell atlas as the normal human reference. Don't you think that common queries will be, I have this data, is it in range in the normal distribution or is it out of range, for example? Don't you think that will be a common query? I'm asking the rest of you. I would expect that those would be
absolutely. I think that and we'll come on to that, because I think that query, especially in the context of disease, is a very natural thing to ask. What cell types are enriched in my sample or depleted? But before we get to that, just maybe if we could keep the answer to this question brief. The human genome has tidied up its assembly for decades, for example, at the telomeres. When we think about a reference atlas, what are we still going to be fiddling with in the context of HCA in 20 years' time? What are we going to be improving? Maybe we'll start with Aviv for this.
Can you repeat it, John?
What are we going
in the end on my Zoom?
Okay. What's the equivalent for tidying up the telomeres in the human reference?
Finishing. Yeah. Finishing. So I think the most notable equivalent is getting to rare frequency events. And a little bit, it's like tidying up the telomeres, and a little bit, it's like getting to rare variants in human genetics. All these analogies are wonderful, but they're never perfect. And I see two versions of them. The first is getting to really just rare cell types, you know, like ionocytes in the lung and other rare cell populations where the proportion of the cell is low, and they would be especially difficult to capture if they're not just a low proportion cell in aggregate across a large tissue, but if on top of that, they only exist in a particular location in that tissue, like the heterogeneity we have in the brain, where something can be rare, but it's also rare in a rare location. In those cases, only as sampling becomes more and more systematic in how we span large organs, would we actually get to those things. So that's one. The second one are transient events. So things that only exist at, you know, particular moments in time. Sarah talked in her part of the presentation on development. That's a great example of a transient event. I showed an example from the COVID atlases, which was about transient events that exist when tissue tries to regenerate. When that transient event goes awry, you might actually capture it, mostly because it gets stuck there. So you see more and more of these cells accumulate. They're like piling up and not proceeding. But when it proceeds in its natural way, it can actually be a very small fraction of what we're seeing. And here is what I actually have hope for the second class, that as our modeling becomes better, even though if we have some gaps in the transitions, we might be able to make better predictions on the fact that something might be there. I think those are the two obvious things for finishing from a kind of fundamental cell biology perspective. The other big one is diversity. Humans are diverse, so are their cells, but especially humans are diverse, and they're diverse in all of these features that we've just talked about and that Shyam highlighted. I think getting to that level of finishing is still a ways off.
Okay, so other people with thoughts on this? Emma?
I agree with Aviv, but I would also like to add that I think that there are data types that we haven't even started to collect yet that I would love to see that might be in the end, where we don't have the methods yet. So I'm thinking protein-protein interactions. That's really what's underpinning all the functions in the cell. And ultimately, we're mapping functions of healthy cells, and we're classifying function and cell states. And there are recent papers that show that if you compare two cell lines, even though they're transcriptomically similar, 50% of the protein-protein interactions are different, while 50% are conserved. And there's a lot of rewiring that is happening among functional units in the cell, among those interactions that are not conserved. So I personally don't think that we will understand the dynamics and particularly these transient events that Aviv mentioned before we can also map protein-protein interactions at a single cell level, and we're not quite there yet. So I think that there will be things like this that will last for at least 20 years to come.
Excellent. Shyam?
Well, Aviv and Emma have just described how hard it is and how far we need to go. I think it would be nice to define a metric of completeness, to just measure how far we've gone. So for the human genome, it's just X%, 99% complete, 99.8% complete, and so on. For us, perhaps a metric of completeness would be like if I take a liver, and I have an atlas. It has so many cell states defined, and let's say 98% of liver cells in an average liver will lie within an expression distance epsilon of my reference atlas. And then perhaps we can say that's, it's a two-dimensional definition of completeness. We have a fraction of cells, and we have an epsilon distance. But this is just a first stab, okay? But I think we need to define some numbers to quantify completeness.
Then Fabian, and then we'll move on to the final bit.
I was thinking, you asked me 20 years. It's like so long. Imagine, was it 20 years ago? Did we even have microarrays by then for short RNA sequencing, right?
Yeah, so there were microarrays.
There were, right? So I don't know what. I'm just fascinated what all kinds of cool assays people come up with in a year, let alone 20 years, right? So I'm sure we'll find all kinds of things that we can be enriching on top of it and sort of add layers by layers for particular fine-grained things. Of course, this will not be then done across the whole thing, but parts of it could potentially transport it across. So I think this fantastic, incredible speed of how biology defines and finds out new things. We briefly just mentioned this developmental atlas on the whole aging question. So I think the whole temporal aspect on that long scale, but also on short scales, I think that will definitely keep us busy for a very long time, let alone then. And that's still the healthy part. So that would be sort of in that vision of what the original HCA claimed. So I think having really this trajectory where we just throw in a single cell and then sort of make those trajectories fully bloom up until we die, I guess that will keep us busy.
So then maybe that's a really nice transition to the very last bit here, and that's about taking some of these disease samples and asking Emma's question, how do they differ from the reference? And I think that if we look back at other large-scale genomics projects, one of the major things that they've achieved is relatively conventional thinking around file formats and how the data are stored, for example. Some of the major things are like FASTQ file formats, VCF files for looking for variants, for example. So when we think about taking a disease sample and then mapping it onto the reference, what does a cell type mutation look like? And how would we encode that computationally so that that information can then be shared with the wider community and other people can build upon that with meta-analyses, etc., without having to go and do a whole lot of redigging into the data? Maybe we start with Seda here.
Yeah, so the obvious one, is one of the things that we've mentioned earlier, which is EQTL kind of things, which is the expression variation linked to mutation, and another one is what you've worked on with approaches like Milo, which are showing shifts in transcriptomes in the neighborhood of a manifold. It'll be interesting to think about what a change, how a change can be quantified in terms of tissue architecture in 2 and 3 dimensions. I think that's kind of an exciting challenge for the computational biology community coming up. You could think of changes in the neighborhood of cell types relative to each other, for instance, quantifying that in some way. So those are the main modalities. At the end of the day, the main sort of measurement modalities for human cell atlas are suspension, single cell, single nuclear genomics, and then spatial genomics of various descriptions. And so then it's changes in those, computed in those modalities.
Excellent. Fabian, I think you were next.
I think from what we sort of discussed before was right that for us, description of an atlas would be this distribution across expression and maybe also a covariate space. So I guess what you would need to do for differential one is then do differential distribution comparison. There's all kinds of ways, people have been using these Wasserstein distances and related things so far. And I think depending on how we project, then we come up with relative cell type frequency comparisons, which is actually something in many cases already explains quite a bit, maybe then this sort of new particular state popping up. But I think as long as we come to agreement what the categories, I think Sarah has this nice statement that categorization is so important and powerful for driving our thinking. As long as we come up with those categories, then we can do differential comparisons of those categories, and that would then really hide a lot, or sort of not even obfuscate, just really deliberately hide, but make it sort of in the background available if they want to users, well, how they can use an atlas. Because very often it might be just really sufficient to say, hey, I see this cell type enriched, right? And sometimes more you want to maybe ask more complex things. But I think in the background, these tests need to be really on the distribution.
Yeah, I really like that difference of distributions. I think that's very useful as a way of thinking about it. Emma?
I actually want to say pretty similar things as Fabian just said, but I'll just simply add, I think also we need better models to embed phenotypes, because it's actually the phenotype that we're comparing, and phenotype can be more than the single cell seq data. So I think that we can do quite a lot on better complex descriptions of phenotypes for comparing these distributions.
Excellent. Very quickly, Aviv.
Just to Emma's point on the embedding of phenotypes, one of the nice advances that has happened computationally in the last few years is that the ability to relate things that are nominally appear completely unrelated, like here's a picture, here are words, or here's a picture, here's gene expression data, has become dramatically better. That gives better phenotypes, but it also gives better explanations of biology. You can now say, oh, that is made of this causes that. These are things that were very difficult for us before without a direct experimental measurement. We now have the possibility of doing that, and having the richness and the scale of data like the HCA, including both spatial and cellular data, I think would dramatically help in moving that forward.
Excellent. And so in the interest of time, we're just going to have one final question for everybody in a quick answer. Given a reference atlas, what's the query you would like to make of it? So we'll start with Aviv.
What cell did I not see in this data?
Excellent. That's a nice short and sweet answer. Seda, you next.
What cell is present everywhere and not inside blood vessels, but outside? Excellent.
Shyam.
I'd say, where do my cells lie relative to this atlas?
So I see a vision of sequencing yourself, Shyam, and now sticking it in, which would be quite hard to do a query for. But anyway, Emma?
I would want to ask how many metabolic cell states are common to all cell types?
Fabian?
What gene dependencies would I pick up here specifically?
Excellent. Those are 5 fantastic and thought-provoking ways to end the session. So I would like to thank Sarah, Aviv, Fabian, Emma, and Shyam. That's been a really enjoyable session.